February 18th, 1903. Deep beneath Somerset, a nearly complete human skeleton was uncovered, hailed as Britain's original inhabitant. For over a century, textbooks and museum displays repeated the same story about who he was. But one fragment of bone and a revolutionary DNA breakthrough shattered those accepted truths. If ancient Britons weren't who we thought they were, what else about our past has been rewritten without us knowing? In the winter of 1903, the limestone chambers of Gough's Cave echoed with the scrape of shovels and the low voices of Harold St. George Gray's archaeological team. Gray, a meticulous recorder of detail, pressed on through layers of ancient sediment, mapping each find with careful notes. On February the 18th, as daylight filtered through the cave's mouth, the team uncovered something extraordinary: a nearly complete human skeleton curled in the earth, bones largely undisturbed since the Mesolithic era. The discovery pulled the attention of both scientists and locals, who crowded near the entrance, eager for a glimpse of what Gray's team had found. Gray's field diary, preserved in museum archives, describes the careful extraction of the skull and long bones, noting the depth and arrangement of each element in the stratigraphy. The remains were quickly recognized as ancient, far older than the Roman or medieval bones sometimes found in the region. Early newspaper reports from Somerset captured the excitement, with workmen quoted as marveling at the idea of Somerset's original resident lying in their midst. Gray catalogued the skeleton with scientific detachment, but the scale of the find was clear. This was the oldest nearly complete Homo sapiens skeleton ever discovered in Britain. The bones, packed and sent for further study, would soon become known as Cheddar Man, anchoring a century of debate about Britain's earliest people. The cave, long a site of local legend, now held a direct link to the deep past, a single body preserved through thousands of years, waiting for its story to be revealed. Within weeks of the discovery, Cheddar Man's bones became a national curiosity. Newspaper headlines branded him the oldest Briton, and speculation about his appearance filled columns and museum pamphlets. Early illustrations produced for the press and for display leaned heavily on the conventions of the era. Artists lacking any genetic evidence defaulted to familiar European features: straight noses, light skin, tidy hair, mirroring the faces of those who commissioned or viewed the work. These renderings appeared in illustrated weeklies and museum catalogues, often captioned with language that reflected the racial attitudes of Edwardian Britain. Terms such as primitive European or ancient Caucasian were common, and some displays even included comparative charts placing Cheddar Man alongside modern British types to suggest a direct, unbroken line of descent. The absence of hard evidence left wide room for projection. Museum staff and popular writers described Cheddar Man as a forerunner of the modern Englishman, reinforcing a narrative of continuity that fit national myths. Early reconstructions, sometimes produced by staff artists with little anthropological training, presented him as robust but recognizably British, his features softened to fit contemporary ideals. These images circulated for decades and shaped public perception of Britain's prehistoric past. The reliance on surface resemblance and prevailing racial theories meant that Cheddar Man's true story remained hidden beneath layers of assumption. The press and museums, eager to offer a tangible origin for the nation, inadvertently locked his image into the mold of their own times, setting the stage for a later reckoning with the evidence. A century after Cheddar Man's discovery, the question of his true origins remained unresolved, until a new approach offered a path forward. In the controlled environment of the Natural History Museum's ancient DNA lab, researchers turned to the petrous part of the temporal bone. This small, dense section of skull, nestled near the inner ear, is renowned for its ability to protect DNA through thousands of years of burial. The decision to drill into such a rare and complete specimen was not taken lightly. Conservation staff, geneticists, and an institutional ethics panel weighed the risks and benefits, debating whether the scientific value justified the irreversible removal of even a tiny fragment. Once permission was granted, the team extracted just 25 milligrams of bone powder, about the weight of a paperclip. Every step was performed inside a clean room, designed to keep modern human DNA out and ancient molecules in. Technicians wore full-body suits, and surfaces were swabbed repeatedly. Blank controls and negative samples ran alongside the real extraction, ensuring that any trace of contamination would be detected before sequencing began. The petrous bone yielded an exceptionally high percentage of endogenous DNA, over 40%. A remarkable figure for remains of such age. Multiple independent extractions and library preparations were performed to confirm the results. The sequencing run, carried out on an Illumina NextSeq 500, produced the highest coverage Mesolithic genome ever recovered in Britain. Quality checks confirmed the ancient origin of the DNA with characteristic patterns of molecular damage and negligible contamination. These precautions and results gave researchers the confidence that the genetic data derived from Cheddarman's own cells, not from those who had handled his bones over the years. The journey from a fragment of bone to a complete genetic readout was now finished, setting the stage for the findings that would follow. Geneticist Dr. Johan Diekman peers over a screen, reviewing the code that translates ancient DNA into visible traits. The data from Cheddarman's genome, run through the HiRisplex S model, delivers a surprise that upends a century of assumption. Two genetic markers carry the most weight for skin color, SLC24A5 and SLC45A2. In Cheddarman, both sites hold the ancestral or original versions of the genes, versions that do not lighten skin. Dr. Diekman explains that Cheddarman had the ancestral versions of all pigmentation genes, strongly suggesting he would have had dark skin. The same DNA scan picks up the HER2-OCA2 variant that controls eye color. In this case, Cheddarman carries the derived form associated with blue eyes in modern Europeans. The combination is striking. Dark to black skin, blue eyes, and dark, tightly curled hair, a palette rarely seen in today's Britain and almost never in earlier reconstructions. These results are not the product of guesswork. The team input Cheddarman's genetic data into a probabilistic model which weighs each variant's effect. The absence of the skin-lightening alleles paired with the blue eye marker makes the prediction clear. Confidence in these calls is high, thanks to strong DNA coverage at the relevant sites. This genetic portrait is not unique to Cheddarman. Similar patterns appear in other Mesolithic Europeans such as Labrana in Spain. These findings force a reconsideration of what it meant to look native to ancient Britain. The evidence now points to a population far more diverse in appearance than once imagined, with traits that defy modern stereotypes. Subscribe for more investigative history content. Archaeological evidence places Cheddarman among the Western hunter-gatherers, a group that entered Britain as the last Ice Age retreated. Around 10,000 years ago, rising temperatures opened a land bridge known as Doggerland, linking Britain to continental Europe. Groups of hunter-gatherers followed herds and rivers northward, settling in newly accessible forests and valleys. Genomic analysis confirms Cheddarman's closest genetic relatives lived across Western and Central Europe, from Spain to the Baltic. Dr. Tom Booth, a Mesolithic specialist, describes this population as highly mobile, adapting to shifting landscapes and climates. Their arrival predates the spread of farming by several millennia, placing Cheddarman's community at the heart of Britain's first permanent population after the glaciers. This migration left a faint but measurable genetic legacy, connecting the Somerset skeleton to a broader wave of settlement across the continent. In 1997, a history teacher named Adrian Target received unexpected news. DNA testing revealed he shared a rare mitochondrial sequence with Cheddarman, connected by a maternal line that stretched back nearly 10,000 years. The match was found after scientists at Oxford compared a tooth from Cheddarman to samples from 20 local residents. While the link is distant, reflecting a common ancestor rather than direct descent, the story resonated in Somerset. Museum curators recall visitors lingering by the display, some searching their own features for echoes of the ancient skeleton. The finding became a talking point in schools and sparked debate about what it means to belong to a place. For many, the idea that a living resident could share any genetic thread with Britain's oldest skeleton brought the story of Cheddarman out of the cave and into the community. Today, DNA evidence reminds us that British identity is rooted in migration and diversity, not myth. As science reshapes our sense of ancestry, the story of Cheddarman challenges every certainty about who belongs. History isn't set, it is rewritten with every discovery. Subscribe for more investigative history content.